So thank you guys for coming. Thanks to everybody on Zoom as well for logging in. It is my honor to introduce Lin Chao. So the first time I learned about Lin Chao, I was a master's student, and I had this really engaged, really ambitious master's student colleague that had found one of Lin's old papers and showed it to me. And then we started digging through all of his papers. And we were both astounded that there was this guy that was at the formation of our field, experimental evolution, and that he had been behind so many of these really cool results that we were really compelled by as students. And so we read through them together. We read papers about early modeling of microbial communities and population dynamics. Somebody is, please mute yourselves. So, and then we read his papers about applying game theory to understand viral genomic evolution, and then also providing a biological example, a biological test of game theory. Then we learned about how all of this sort of fitness landscape research he had done and how he had applied fitness theory to make, fitness landscape theory to make predictions about populations and to test them in the lab and develop, really sort of some of the main ideas behind that kind of theory and predicting trajectories of evolution through that theory. And now today he's doing this, amazing research on aging in bacteria, and again, developing theory that makes testable predictions and going to the lab and actually testing those predictions. So the second time I encountered Lin, and was actually the first time I actually met Lin, is when he showed up a month early to a conference at Michigan State. So he's got the absent-minded professor stuff down, certainly. But he completely recovered because he gave what I thought was the best talk I had ever seen at the time, and certainly is one of the outliers and one of the best talks I've ever seen. So my third experience with Lin obviously is moving to UCSD and now getting sort of day-by-day interactions with Lin, and that's such an honor to have. And I've learned sort of a lot more about him personally. And so one of the aspects of his career, his life trajectory that I think a lot of people don't know about and overlook is that Lin grew up two immigrant parents and you can correct me if I'm wrong about this, sorry, in Brazil. We just learned a couple of weeks ago at the EBE seminar or EBE happy hour that he used to play soccer in the favelas. And then in the 1970s, he left there to go to Cornell University. And so that's a similar arc as mine. And I always make a big deal out of being a first generation student and an inner city kid going to this Ivy League university. But I can't imagine how many more barriers that Lin actually had to go to Cornell University as well. So it's an absolute honor to introduce Lin today. His students have all gone on and his trainees have gone on and are now leading research roles at Yale and Brown and so the Ivy Leagues and also major European institutions. And so so many of his trainees have done so many great things, and that's because he invests so much in his trainees. And so he's really made a huge impact on our research and our field. And let's welcome Lin.
Sorry if I look ridiculous, but I need a long leash in life. Thank you all for being here. I'm not going to give a big introduction. I'm just going to tell you a story, what I've done the last 10 plus years. All stories have a cast of characters, so let's just dive into that. Camila, major colleague. I've been working now with her for 20 plus years. She brought to the lab all sorts of microbial approaches and thinking, microscopy, and at the end, I will actually use some data she collected for her PhD thesis, which looked at the relationship between ribosome numbers and the growth rate of a cell. Okay. Other people have been big influence on this project is my postdoc, Martin Ackerman. I'll talk about how he affected it. two graduate students, Audrey and Chao. The students here who've been a little bit more than a year will know them. They're all doing very well. Chao is teaching in China, and Audrey is doing a postdoc in Berlin. Audrey will be in the job market. That's all I'll say. Undergraduates who've done masters or rotations in the lab, you know how important they are. They collect preliminary data that lead to projects by students, us, grants, etc. Very, very important role. And then faculty that I interacted closely with, I interact as much as I can with faculty, but these are the ones who have really had big effect on the progress of this project. Scott, he is my favorite sounding board. And then all these physicists here who are my antagonistic sounding boards, okay? But you know that sometimes in life, having a strong challenge is what you want. And I gotten it from them. I'll talk about that. And if there's people gonna report back to them, it is in no way disrespectful, okay? We disagree, we agree, but that is what makes science go. And I'm very happy to have worked with them, and they have made very big contributions. And then funding NSF copies is an important one. It is a funding that from Brazil that unfortunately has dried up but may come up again. And it brought Audrey and Mari, two students from Brazil, to UCSD. Okay, I came here probably 20 plus years, and I came, as Justin mentioned, looking at mutations. Are mutations on the average good or bad? What's the effect of linkage? What's the effect of recombination? Is there interactions that are additive and non-additive, pleiotropy, epistasis, et cetera, et cetera? That was what our lab did. And then one day with Martin, we had a revelation. Well, we realized that we have been talking about mutations, and you can think of mutations as being damage to DNA. But another source of damage was happening in cells. And these are non-genetic damage, for example, oxidized proteins. So here, we're thinking of an E. coli, a cell, has damaged proteins. What is that cell to do? Well, you'll say repair it. Okay, but repair has a cost. What happens if the net benefit of cost makes it such the repairing is gives you no advantage? Then you're stuck with it. So if you're stuck with it, what should a mother bacterium do at division? Parents love their children equally, so you should give everything to your children in equal amounts. But maybe you have a favorite, and it should be asymmetrical, and you'll give more damage to one child because you like it. Sometimes in life, for us, we encounter a situation in biology where no one has done a modeling. The rules of genetics allowed us to do this type of work. But what were the rules of transmission for non-genetic material? No one had ever done it. And that is like this moment, right? It was like, okay, we can model this. So that's what Martin and I did. And we went through 2 generations of models. I will walk you through and give you only the last generation, partly because that's the one that gave us the most useful results for testing. Before that, however, I have to give you some nomenclature, and that will be discussing what is known as the polarity of bacterial cells. So I draw for you here a bacteria. Forget the numbers for now. You all know E. coli gets long, then divides in the middle. So it'll be cleaving right here, and then 2 new cells will form. These poles here were just formed. So that's why they're numbered 0. They're brand new, so we call those the new poles. The distal poles, which is probably a better description, however, they've been around from the last generation. So those are called the old poles. So all E. coli, if you ever reach in and you could grab an E. coli, one of its poles will be new with an age of 0. Always. Every E. coli will have that because they were born by cleavage. The other pole will have a certain age and we won't know what it is. So I arbitrarily chose a 1, but I could have put in 1 million, and this E. coli would date back to, you know, way back, okay? Origin of the mammals, okay? Something like that. So if we take this to be 1, 0, you know that if I divide here, these are 0, 0, but this used to be 1, so now it becomes a 2 because it is 1 generation later. The previous 0, which is right here, will become a 1, and so forth and so forth. The middle poles will always be mu, and the distal poles will always be called 0. So that's the first point. Second point is, if I have a mother cell with an old and a new pole, when this mother divides, the daughter that inherits the old pole is called the old daughter. Okay, so that's another complication. They're old and new poles, and they're old and new daughters. The old daughters get the maternal old pole. But then old daughters will then become mothers, and they're called old mothers. Okay? And if we follow a lineage, notice mother, old daughter, this is another old daughter, right? Because it got the previous one. So this is kind of the old granddaughter, if you wish. Okay? So this would be called OO. This is new because it got the new pole, and it gets the new pole again. So these are just ways that we think about it, and you need to keep that organized because I'll be jumping back and forth between old and new mothers, old and new daughters, and old and new poles. The idea that Martin and I had did not come out of nowhere. It came out of a key paper that came out in 2005, and it was published by the French group in Francois Tadet's lab. Eric Stewart is the first author, and they basically showed in this paper and in a later paper that the old daughter grows more slowly. So the distribution of damage, which they thought was the causal effect, was asymmetrical. And as a result of that, the old daughter gets more damage and grows more slowly. This graph kind of gives you that. Over here is growth rate, how fast they grow. And the red from now on will symbolize old daughters, and the blue, new daughters. The blues tend to be on this end, which is the higher growth rate. The reds are on this side, which are the old daughters, and they grow more slowly. On this axis is the fluorescent value for a chaperone that's associated with aggregates of protein damage. So more fluorescent, presumably a larger aggregate. So the old daughters, which are red, sometimes will have very large aggregates of damaged proteins, and they grow the slowest. There's a negative relationship here, driven primarily by the red dots, right? The old daughters. They have a range in how much damage they have, and that gives a negative correlation with the elongation rate. The new daughters tend to be over here, very little damage, and they do have a range in growth rate, but they tend to have very little measurable damage. This graph is collected by physicists. Physicists don't like to infer causality. They think that everything's a correlation. Therefore, the axis is down here, growth rate. I would have flipped it around and put damage down here, helping me to think that damage is causing elongation. You can think it any way you want, okay? But that's a premise that I'm trying to work with. So we went through a bunch of models, and the final model is the one that I will talk about. It came out in PLOS Genetics. And the reason why this model became the final one was we came upon a realization, and that is the following. Unlike damage to DNA, which are mutations, damage, non-genetic damage like oxidized proteins, which can be inherited, right? Because if I'm passing damaged protein to my daughters, there's a heritable component. It's an epigenetic process. You can think of it that way. But this epigenetic process manifests the deleterious consequences of that damage immediately. Science imitates life. I cut off my finger on a table saw to prove that. And I suffer the consequences immediately. It isn't the next generation. There's no phenotypic lag. So we had to put that phenotypic lag into the model, and that was done in the final paper. So I'll walk you through that. So here's the model. KT is the amount of damage a cell has at any point time t after birth. Okay, they're born at t equals 0. So K0 is the amount of damage the cell was born with. However, as the cell lives and time goes on, more damage will be picked up spontaneously by simply growing. That rate is lambda. And that's the analogy to mutations. If you do mutation models, you'll have a term called mu-t, which is the amount of mutations you have with the passage of time. Very quickly, we call this the equation of life. This is the amount of hurt you have at any point in your life. K0 is all the baggage that your parents gave you. And lambda t is the insults that life will throw at you through your life from birth to death. Okay. With that, we can now model a cell, and we think of a cell as having to do something in order to divide. For example, double in volume, double in length, or maybe make a certain amount of protein. We do not know exactly what triggers the division of E. coli, but it must do something. So we simply call that capital P, and we write an equation for dP dt, the rate at which that product will build up. We scale it to 1, so that is just an arbitrary unit, but we put a penalty and say, look, the amount of damage will slow it down, so we simply assume it to be linear, and you can argue that, but first pass was linearity, and the rate will drop depending on how much damage I have. So I can rewrite that. KT equals that. So dP dt equals 1 minus K0, etc. We can then integrate this, simple calculus. You get a solution as a quadratic equation, but that'll tell us how much product I have. I said when I have a certain amount of product, I'll divide. We call that pi. So when I reach the pi, I'll divide. So when PT equals pi, I divide, and that time is important because that's a time of division or the doubling time. That's how long it took the cell to double and divide and produce me2. So this T becomes capital T. So I now have an equation where if you tell me how much I'm born with, K0, I can tell you when you will divide. That's important because if I know how long it'll take for me to divide, I can take that value of capital T, the doubling time, and put it back in here, and I know how much damage the mother had when it divided. And that's very important for a simple reason. That amount, K, as a function of capital T, the doubling time, is the damage that the mother will partition to the daughters. So the partitioning is done where A is given to one daughter and the other daughter gets A minus 1 minus A. If A is 1/2, I'm equally loving to my children. They both get 1/2. However, if A is less than 1/2, and we could actually make it bigger than 1/2. No, we can't. It has to be less than 1/2. One daughter will get more and the other will get less. This then allows me to calculate, given the doubling time of a mother, I can tell you the doubling time of the daughters, because if I know how much damage they were born with, I just go through the previous equations and get the doubling time for the daughters. That's what we want to do in evolution. We want to take a parent and predict the offspring. Because if I can predict the daughters, I can predict the granddaughters, and I can go on forever and do a population and then study evolution in silico. So that's what we did. People ask me, why is it that asymmetry of distribution of damage, asymmetry of growth rates that results from that damage, hypothetically, is beneficial to the lineage? Why is it that asymmetry has evolved? The claim is that one daughter will grow faster, but the other one will grow slower. Is that good or bad? So they say, why has evolution favored this? And the models come up saying that the fitness of the lineage although one daughter is slower, but the other one's faster, gives the lineage a net advantage over symmetry. And if you need intuition there, this is what I throw out. Take $1,000, put it in a bank, get 8%. You can calculate how much you'll get. However, if you could take 500 and put it in a 6% account and the other 500 in a 10% account, note the average of 10 and 6 is 8, just like before. After 1 year, you'll have the same amount of money. But after 1 year, you'll have more money if you do it this way, which is why you're supposed to diversify your portfolio if you're managing your retirement accounts, okay? Because the faster growing account at 10% will overtake the 8% one. In fact, you can make this 0 and the 10% will eventually overtake everyone, okay? The bacteria are managing their retirement very well, okay? And the lineage benefits through this asymmetry. The model showed that in theory, you could get a higher fitness for the lineage, but many other details emerge, and I'm going to walk you through that graphically. So the way to think about this is now imagine the doubling time of the mother, which is the input, right? And the output is the doubling time of the daughters. There are 2 daughters, one old, one new. Old daughter is always red, new daughter is always blue. And the predictions, basically, if I have the mother of a doubling time of 30, I get a doubling time of something for the old daughter, and the point will fall on the red line. Important thing to realize is that the red line is higher than the blue line. Why? Well, this is doubling time. So if you grow fast, you have a short doubling time. So the red one, the old daughter, has a long doubling time, meaning that it takes a long time to divide, lower fitness. So yes, the old daughter still has a lower fitness, and that's represented by the red line. And this is the mapping. Give me this mother, I can tell you what the daughters will have. Likewise for the blue. First important event is that both of these lines have a slope less than 1, and they cross the diagonal, which is the identity line. That means that these intercepts are equilibrium points. They're stable equilibrium points. If the line was the same 45 degrees identity line, those of you who are technical about this, you'll get random walk. If you have less than a 1 slope, you'll converge to that point. And you can do that graphically in the following way. Think of a mother with a doubling time of 30. She is slow growing. E. coli likes to grow around 20 minutes. 30 is a slow time. This cell is loaded with damage. The daughters, however, will do better. Why? Because growth is adding new tissue to the cell, so it dilutes the damage. So that's why the daughters will have a doubling time. This is the old daughter that gets more damage. Its doubling time will be less than whatever 30, right? Because 30 will be up here. So this one's like, 26, 27. So that's the daughter. Well, the daughter becomes the mother of the next generation. So we go over here and then we come down. So this is where the daughter is. And the granddaughter then will be here. And we follow the lineage. You will march through time, through division of the lineage to this point. You will not go beyond it, because if we go beyond and start off over here with a daughter that has very little damage and having a doubling time of 22 minutes, it will pick up damage. Imagine that daughter has 0 damage. It'll pick up damage. So this mother's daughters will be worse off, and they will then get a longer doubling time, grow more slowly, and then converge towards here. So if you come from both sides, that becomes a stable equilibrium point. If that is true of the old daughter, it is also true of the new daughter. So both of them are stable equilibrium points. E. coli turned 200 years last year. E. coli K12 was first isolated in 1922. There's no celebration. I don't know why. But for 100 years, we've been looking at this culture every morning, and we never realized that there were like 2 subpopulations in there. one of the old daughter, one of the new daughter. So next time you look at E. coli culture, think of that, okay? Does that mean that the culture is separated into two distinct populations? No. And the reason is, imagine that I was an old mother at this equilibrium, which means that I had a doubling time of, say, what, 26 minutes? That mother, when it divides, its old daughter will fall at 26 minutes, but the new daughter will be down here. The new daughter then, through division, and must create a new daughter lineage that will converge here. And likewise, new mothers here will produce old daughters here that will, through lineage replication, will converge there. So we should really add arrows. And the important thing to realize is that this is aging, this is rejuvenation. I'll try to link this to you later on, okay? But for now, this is just E. coli. You can say, well, that's aging, that's not real aging. But we can talk about that later, okay? But this is slowing down as the cell reproduces, and here the cells are rejuvenated until they come over here. They're losing damage. If this is true, it means that E. coli in a LB culture in the lab is immortal. Of course, if you autoclave it, it dies. But short of that, and you keep on passaging, it is, under these conditions, immortal. It is not dying physiologically through accumulation of damage. It comes to an equilibrium and stops and stays there. Cell lineages are immortal. Equilibria prevents immortality. Something to appreciate, that this aging and rejuvenation process happens only because there's asymmetry. If E. coli were symmetrical, the two lines will converge to one, right? The two daughters are identical. Notice that if the black line described E. coli, there'll be only one equilibrium, and everyone would stay there. There'll be no rejuvenation or aging. Everyone has an equilibrium with some damage, but they all be equally damaged. It is only with asymmetry that the lines separate and I get aging and rejuvenation. That's very important. It comes with the asymmetry. Is mortality possible? In the model, it is. In the model, we had a parameter that was lambda that reflected the amount of damage that an E. coli experiences as a function of time. Well, if we do that in the model and start cranking up lambda, the effect is that the red line, the old daughter line, starts having a steeper slope, becomes extremely nonlinear, and bends upward. In other words, this intercept is no longer crossed. The equilibrium has disappeared. So if you were to do the little arrows I did you earlier, it's going to go up and go up until the doubling time becomes infinitely long. In essence, E. coli has stopped dividing. The population or species will not go extinct because the new daughter equilibrium still exists. And that equilibrium will seed the population and keep it going. However, the lineage descending from here, the old daughter, right, is going to come here and go. So the old daughter descendants from this equilibrium will eventually die, kind of like us, right? Our children will do well, we die, they will then age and die. I'm just giving you a way to look at it, how I'll try to bring it together. So, immortal, but mortality is possible. It is only a function of the amount of damage they experience. I'm just checking time. I'm halfway, okay? Halfway. Okay, the model. It's a complicated model. It took us a lot of work to develop, so I spent so much time. But I feel like without the model, you won't appreciate what we're trying to do. So we did this model. We're very excited. We thought we had solved everything. And there was a biophysics conference here at UCSD organized by the physicists. We show up with our model. We say, we're going to collect data. We're going to write an NSF grant. We're going to publish papers. And we're all excited. And I run to Terry Hua and he says, Lin, it's all an artifact. And he says, a paper just came out showing that the results Tadej got do not hold up. And I said, but Tadej did statistics, everything was significant. But the difference, according to Terry, was that the sample size for Tadej's paper was in the hundreds, at the most thousands, and this new paper had sample sizes in the tens of thousands, if not hundreds of thousands. The paper was by our new colleague, Suk Jun. This is a very famous paper because it introduced what is known as the mother machine, which is just a very small test tube carved into plastic that's the width of an E. coli cell. The cell is in here, and as it doubles, the daughters are pushed out, and eventually media that's also diffusing and feeding it flows away and washes out the waste and the daughters. But you can image this cell down here for weeks. In a good run, you can get 2 weeks or hundreds, if not 500, if not 1,000 generations. And these are 1 micron big, so you can line up thousands of this in a millimeter, and you can collect hundreds of thousands of measurements. And the result gave this, which is constant growth rate with no aging. And the title was called Robust Growth. They didn't want to go head on and say that aging was an artifact and did not exist. Why? Because Francois was a co-author. Okay? But that was not reinforcing, because if Francois could not figure out why his data was flawed and stand up, could it be that even he thought that it was not valid? And that's what Terry was telling me. So it was not a good weekend. I went home, downloaded Suk Jun's paper, and read it nonstop for a week. There had to be something. And there was. This is the mother machine. A cell goes in and starts the process. You can ask then, will the pole that goes down be an old or new? 50-50 chance, right? So I draw it for you, so the new pole goes down. Cell divides, cleaves right here, new poles are made. This becomes an old pole. But this is a new mother, right? Because it came from an old pole of the cell that first went in. So this, we don't know what it is, but we know that this is a new daughter, therefore a new mother. But that is true only for 1 generation, because the next generation, I elongate, cleave over here, and the old pole is stuck pointing down from then on. Which means that after 1 generation, you can say with certainty that the cell reproducing at the bottom of the mother machine is an old mother. So the constancy that Suk Jun saw turns out to be the strongest evidence at that date for our model. It is simply demonstrating the existence of the old daughter equilibrium. This is what the mother machine had documented. It was robust. It was an equilibrium as we had predicted. That was a big relief when we saw that. So how can we test further? Well, we want to now take this and follow the diagram that I gave you. This will make a new daughter. It should come down and eventually convert to another equilibrium, which was the new daughter. But we can't do that. And the reason why we cannot do that is because the mother machine, the new daughters are being washed out, and we can't track them. And the currency now was sample sizes in the tens or thousands. So how do we get 1,000 new daughters? Physicists come to the rescue. Jeff Hasty and a student, Ottavio, had designed another microfluidic device where the cells were held in a monolayer, media flows on this side and feeds them. But the population now is in the hundreds of cells, and the new daughters have a residency time that allows them to survive long enough for us to quantify them. So we call this the daughter device. So with the mother machine and the daughter device, we could collect parallel information. And the goal now was to see if we follow daughters, do we see a new equilibrium? So from the daughter device, we get this. It is noisy, okay? And noise will come in at the end. I'll talk about it. But the daughters that are old will have a doubling time of 21 minutes or so, and the new daughters, 19 minutes. The blue tends to be in the bottom, the red on top. A better display of this data is to do what I did you earlier, which is the mother's doubling time and the daughters, and we track a lineage. And the red line is circling over here where the equilibrium for the old daughter is, and the blue is circling here where the equilibrium of the new daughter is. We took this to represent that the model was making the correct predictions that was being verified. We can do other tests. We can ask, when an old daughter is produced by the new daughter, does it go to that equilibrium as we anticipated? And it does. So here's the trace of an average of many populations. New daughter produces an old daughter, the lineage goes up to that equilibrium. Likewise, when old daughter produces a new, it comes down to this equilibrium. How do you quantify stability? And this is, again, we went to Massimo, who has left but was then, and he helped us derive this through. I had said that what matters is the slope. That is true, but you also have to consider the stochasticity of the variance or the noise. So basically, you want the noise of it's noisy, right? It's not at the equilibrium like at one point. It's like jumping around. That noise will have a variance, okay? So for this point to be stable over long periods of time, the criteria from a physicist's perspective is that the slope has to be less than 1, and it is right here, 0.347. But the summation is m squared plus sigma squared, so the variance, and that sum has to be less than 1. We can calculate that, and we come up with this value, 0.125, so we're safe. And this system is stable as we had claimed. The other moments also come in, okay, skewness, kurtosis, but they become weaker and weaker, and so they don't have an effect, and these are the dominant terms. But what Massimo was able to do is calculate this for us, which we thought of intuitively, but didn't know how to quantify. How about this prediction that if you increase lambda, the damage rate, it'll become unstable and the old daughter will die? We thought of using a damage agent. Hydrogen peroxide is an obvious one, but bacteria can detoxify and break down hydrogen peroxide, so there's a double problem. So then we realized that we always told, don't shine your bacteria too much because the light will kill it. Well, in that case, let's use that to kill them. So that's easy because you're on the microscope, you type in how much light to shine, and now you have a variable to control. So 0 light, these 2 points you've seen before. So then we start exposing all the way up to 3 minutes. And if you follow first the new daughter equilibrium, it becomes a little bit longer, but not by much. So the new daughter is hanging in there very happily, but the old daughter is not. It starts experiencing longer and longer doubling time, 30 minutes, and at about 1.5 seconds of light, it stops dividing. Our observation window is 200 minutes, but after 200 minutes, we understand we would like to follow it until it doesn't die for a year, but we can't do that. So we took 200 minutes as a representation that it had stopped growing. Uh-oh, computer is unhappy. Oh, okay. This is the same data showing to you the spiraling at 3 seconds. 3,000 milliseconds. And the slope becomes higher as we had anticipated, but the noise also becomes bigger. And eventually it shoots up, and that's where we had that 200-minute point, okay? Just so that you can have a reference, this is the data I showed you earlier, but I've compressed the axes so they now scale to the same intervals. So here, sigma squared is smaller. Here now sigma is much bigger. So we can do the same stability analysis. And what you'll find is that here's the slope. It used to be 0.3, right? Now it shoots up to almost 1, but doesn't break 1. But the variance is increasing. The noise, the stochasticity is increasing. And when you sum them, the old daughter breaks 1. And that's the one that goes extinct most often. This is with light. We also did it with streptomycin. Streptomycin causes a mistranslation of proteins, then they misfold. They're like damaged proteins. This is more like the model. Here's the slope, and the slope just increases at higher concentrations of strep, and both of them die. If you throw in here the noise, it contributes a little bit less, but the final effect is that it is always the old daughter that has a higher probability of going extinct. We also done it with heat, and that result is more like the light result. Streptomycin is the one that fits our model the best. So I'll try to hop through some of the final results. What's the cause of the asymmetry in the daughters? Damage. I showed you this graph suggesting there's a correlation, not necessarily causal, but there's a correlation between the amount of damage and the elongation rate of the cells. I just want to indicate there are people that contest this, but there's also people who think it's correct, me being one of them. So I want to keep on trying to analyze it to see how can we challenge it to figure out whether it will fall apart or not. So our attempt to do this was going beyond and asking, so here's a movie. See, right here, it begins with nothing and you see a glob growing. That is the aggregate being represented for you with a chaperone that takes damaged proteins and associates with the aggregate. So if you wait, it will pop 0, nothing grows, and it's always in the bottom pole or the old pole. You'll look over here and you'll see little flecks of green flashing once in a while. Those are the new cells which were born with no damage, or at least no detectable one, and you don't see them here, but then here they get big enough and you see it for a moment. So old pole has it. So the question then is, could the old pole, because it has a clunk of aggregate, have space that's no longer usable by ribosomes? Therefore, that reduction in ribosomes causes the cell to grow slower. And we have the parameters to measure that. So what we did then was to do a study of the distribution of ribosomes inside E. coli cells using a reporter, fluorescent reporter for ribosomes. Just as a heads up, the dark purple colors are high values, and they're occurring in the middle where the division is, which will lead to the new pole. Okay, that's what we're going to quantify for you. So this is a complicated slide quantifying it. I'll just point out this and then I'll simplify for you. Over here is time, birth, second quartile, third quartile, and division time. So this is a cell's life, born, second quartile, third, and then division, or we call death. Over here is the distribution of ribosomes through this reporting system in the new pole of the cell, second quartile length, third quartile, and this is the old pole. So the cell is sliced this way, okay? 4 slices, okay? This is the data. You don't have to really look at it all. Focus on the following. This is the distribution of ribosomes in the old pole of a new daughter, because it's blue, at the time of division. And I just want to focus on division because division creates the daughters, and that's where the asymmetry arises. Well, if you look at here, it's very symmetrical, right? This kind of looks like that. These two match. It's like a mirror image. meaning that a new daughter, when it becomes a new mother and divides, the daughters are going to be very similar. The asymmetry arises here in the old mothers. If you notice, they have less ribosome at the old pole at the time of division. So a graphical representation would be this. This is how a new daughter, when it becomes a mother and divides, the distribution of ribosomes is pretty uniform. This is what happens in an old daughter who, when it becomes an old mother, the old pole has fewer ribosomes. So the question is this reduction in ribosome explainable quantitatively by the presence of the aggregate? We know how big the aggregate is. So if we can calculate how many ribosomes should be displaced, we should be able to predict what their growth rate is going to be. So actually, we're going to do that, but backwards. So this is Camila's PhD thesis. We know how fast the cells grow. So here they are in our hands, okay? If we go up, then we see that this one has so many arbitrary units of ribosomes. The old daughters will have this. That translates to a reduction factor of 0.076. We know how many ribosomes are in a volume of E. coli. So that is about 0.78 micron cube. That's the volume of ribosomes in E. coli cell. We want to reduce this by this factor and ask how much volume has been displaced. How much volume of ribosome has been removed. So we multiply 0.076 by 0.79, and we get 0.06 micron cubed. How big is an aggregate? The best estimates measured by diffusion rates is 0.08 micron cubed. So we're in the ballpark. The volume of our aggregate displaces ribosomes approximately the sufficient amount to cause a reduction in growth rate between old and new daughters. This is where we are, so I'm going to wrap it up by giving you a way to think about this in relation to multicellular organisms. We've been raised to think of bacteria, and we always draw it this way. Mother, two daughters. I say that they're asymmetrical, but we still draw it this way. What I want to do is suggest that the proper way to think about E. coli is to draw them this way. Mother, old daughter, new daughter. But the old daughter is just a continuation of the mother. And the mother is keeping damage in herself so that she can make a daughter that's rejuvenated. If that's true, I can push time forward and think of here a mother that has very little damage. She makes a daughter, picks up more damage by keeping a larger share, like the old daughter would do. And with time, this lineage, which is really the mother over time, is picking up more and more damage. It may come to an equilibrium and not change, or go to a point where it crashes and causes death or mortality. Meanwhile, this mother is basically spitting off daughters, young daughters, new daughters, which have less damage. This is how I think about E. coli now, and it parallels how we think about multicellular organisms. So here is a young mother mouse. She has a baby, picks up some damage, and then over time, she'll accumulate damage and eventually age to death. You may ask, does that mean that multicellular mothers are asymmetrical? Yes. We reproduce by making a single cell zygote. I know that everyone has their favorite hypothesis why a single zygote exists, but I'm throwing in another one, okay? A single cell zygote, whether through adaptation or a spandrel, and that's a word that I'll just throw out and let you work it out, results in the fact that we are extremely asymmetrical, because this finger will never be in my son. And that's because my son grew up from a single cell zygote. In other words, the asymmetry that we experience from the point of view of reproducing is nearly 0 to 100%. We have 1 trillion cells plus, right? So if we make our children from 1 cell, it is almost like the child gets a brand new fresh start with fresh tissue with 0 damage. Of course, that's not true because some will, but the intent is to make it with 0 damage. So the asymmetry that characterizes multicellular organism is 0 and 100%. And that's the only difference between us and E. coli. So I gave you this graph, and you can ask, what does multicellularity look on this graph? Well, first, perhaps multicellularity is so demanding and asymmetry is so much higher that lambda got high and asymmetry got more extreme. As a result, this line curves upwards and therefore does not cross the diagonal. Therefore, that's why we are mortal. How about the blue line, which corresponds to the new daughter, which is our offspring? Well, if asymmetry is 0 and 100%, that means every child we have is born free of damage, which means that they must have the same fitness, which is what this graph really represents. So if we can talk about fitness in our children, basically they're all equally fit, which is to say that that line should be horizontal. It doesn't matter what age, how sick a mother is, that child should be equally healthy. Now, you could say that's not true, and in some organisms, it is not true. It's called a Lansing effect. If this line has a slope that's greater than 0, that's called a Lansing effect. So the mother's age affects the child. That was first documented in rotifers. There's some claim it happens in Drosophila. Human data is less available to look at that, so it's not as clear. This is basically then the last message I have. And this is a summary slide. I'm not going to read it out. I said everything I've said here. So I'll end by saying, for at least the way we look at it now, it is kind of like aging is a price that a mother cell, a mother E. coli pays in order to have a healthy daughter. If that applies to us, that's a way that I would like to keep on thinking. Thank you. Chris.
It's just an amazing example of frequency of other patches. You can't click back again because everything's piling up, so they get rid of it. But there is a way to do genetic combination. And E. coli has a variety of different ways of doing that. If you look into how your bowel system can be circumvented or short-circuited by just the introduction of a small amount of genetic exchange.
First of all, everything I said is not genetic, it's epigenetic. And the proof of that is that a very sick E. coli full of damage can make a new daughter that's rejuvenated. So it cannot be genetic. Second is, I understand what you're saying. And all this will bring in what we evolutionary people call Fisher's fundamental theorem of natural selection, which is mixing things to create variance. The irony is, if we do what you want to do and recombine, we'll make the daughters more similar and make them symmetrical. And that's no good. Now, we'll live longer, right? So what you're saying is that, yes, we made an attempt to evolve, because we're experimental evolutionary biologists, in the lab, a symmetrical bacteria. The experiment is extremely difficult, and we failed. Okay. But a symmetrical one would live longer because it doesn't accumulate damage to the same degree, right? Because instead of keeping it, I'm saying, hey, children, take it, right? So what you're saying is that if we want to live longer, I should have given this finger to my son. That's what I got to figure out. I got to give my skin to my son, right? But we don't want to do that. So we're stuck getting old. That's my view. And bacteria are the same way. So, of course, what you're suggesting, I think is, I see it not so much as a way to cure aging, but to show that the ideas are correct. That if I made a symmetrical bacteria, which is analogous to recombination, because recombination mixes, right? And I then make it Poisson distributed, right? The bacteria will live longer, but they're gonna be less healthy and have a lower lineage growth rate. That's the damn thing, right? If we want to live longer, we can. Mycorrhizas Drosophila live longer, but their early fecundity is low, so we got to pay a price. A less vigorous youth will allow us to live longer. Sergey.
So, so one correctly, the other, the young, I mixed up the terminology, the blue line, right?
Yeah.
The equilibrium never disappears, even if you inflict just a lot of damage.
Of course not. That's the thing about modeling is that you change the parameter and you get what you want. And if you increase lambda very high, eventually even the new daughter, yeah. And we did that. So interesting result is that we wanted to test but never got to test was that if you put antibiotics like strep, you've killed the old daughter first, right? More will eventually kill the kill the old daughter first. It'll kill the, well, that's how antibiotics work. You got to erase both equilibrium.
So, fact that the so if this division was symmetric, the kind of extinction would come at a lower threshold of damage.
Yeah, yeah. And so in that one paper where we showed is that if we get antibiotic with increasing dosage, as you suggest, a symmetrical one will survive better. Asymmetrical, yeah.
That could be one other explanation for aging.
Yeah. Yeah. Go her first, and you. Go ahead. Well, repair is big, right? I mean, we got Randy Hampton, right? Repair is a big issue. But the problem for us when we do this, we don't know the cost of repair. If we throw in repair into the model and say, well, we repair evolve really high rates, of course, it will repair everything, and then there's no need for asymmetry, nothing. So I always say the fact that E. coli is asymmetrical, it necessarily implies that repair can't do the job. Repair is doing the best it can, but can't solve the problem. So the asymmetry is what we call doing the best of a bad situation. The best situation is to repair everything. But given that bad situation, the asymmetry is how they have dealt with it. Yeah.
Rather retaining the damage, we want to flip the
So there is evidence for neuronal stem cells that the stem cell line doesn't keep the damage. The damaged cells become the determinate path, and they go do something. but the stem cell line is protected. And right, stem cell lines are protected from mutations, right? If there's a mutation, you're kicked to the other way. And if you have more damage, you also are kicked the other way. So it may be more universal, this process. There's a question there. Yes.
Yeah, great talk about that. So this reminded me that in some bird species, so sort of going back to multicellular organisms, there's an elder and a younger chick, depending on who hatches from the egg first. And sometimes the elder will kill the younger for food, especially in times of famine. And the parents will sort of watch and just sort of let it happen. And I'm trying to think, how do I link this back to the sort of asymmetry perspective?
So my first reaction is that's a conditional case, which only kicks in when conditions are bad, right? So when conditions are good, you wouldn't do that. The difference is that E. coli is doing this all the time because conditions are bad internally due to spontaneous rate of damage because they have to respire oxygen, right? Oxygen is the breath of life, but it is also a huge killer, right? Oxygen is one of the most corrosive things in the world. And your body has evolved incredible machinery to prevent oxidative damage while using oxygen to do the job that it does, which is to respire, right? Yes. What do you mean by that?
These things.
So let me recast it in the language that I think I get from Terry Wow, okay? Self-organization can explain a lot of this. So yes, okay? And Terry and I have gone through this a lot, okay? So where I win a little bit with Terry is that one is that evolution is going to fine tune it. Because I can believe in but I cannot believe that it self-organizes perfectly for every organism in the world, right? So there's a lot of fine-tuning and adjustment. Now, is evolution nothing more than capitalizing on self-organization? That's an important point that needs to be debated, right? So the model that is using right now for how these aggregates form is that they're misfolded proteins. Misfolded proteins open up and show their, I always get confused, hydrophobic amino acids, and they stick, right? So they naturally form clumps, right? So now comes a chaperone that tries to fix this, right? Well, but that chaperone then is also a good jumping point to evolving a molecule because repair is no longer possible, and take it to the clump and boom, right? So the chaperone, which was trying to fix it, evolves to take it over, right? So this is, it could be that the first thing was simply this crazy thing that misfolded proteins stick. And the old pole will have more of it because the DNA forms a filter in the middle and big clumps cannot go through, only little clumps. So you're going to have an aggregate on the old pole. So that's a model. But then there's fine tuning. But then you'll say, well, why did the internal amino acids in a protein become I always get confused, hydrophilic, right? Well, you say, well, that's so they stick together. Well, then I say, no, that's so they clump and give me asymmetry. I don't say that, okay? But I could on a day arguing with Terry, okay? Yes. I guess last question, okay.
Things.
So this is a good plug for Audrey, my student who's in a postdoc and looking for a job. The project that she is initiating is what that we should have realized and done a long time ago. We try to track damage, right? And we use GFP and fluorescent to do that. What they're doing is constructing a damaged protein that's not natural to E. coli. So they're using the lambda repressor. And use that to see whether it ends up at the damaged protein aggregate, and then follow this whole thing, because the aggregate disassociates and little pieces will diffuse out, and then they glue back, and then that's how a clump forms at the new pole. So those answers hopefully, and then a prion is something special, then that's another complication that I don't know how to think of.